In this lecture, the regulation of kidney functions is going to be considered, taking into account that we are approaching the end of the course of physiology and completing studying the body functioning. It is necessary to sum up the knowledge on regulation of various constants of homeostasis the kidneys are involved into, and finally integrate the understanding of it into your general system of knowledge in physiology. Thus, in this lecture, we are going to consider again some items that have been already studied, but now we are considering it at the higher integrative level. Most important regulation of kidney functions is by hormones. And main hormones that regulate kidney function operate in the distal tubules, in particular late distal tubules and collecting ducts, where they stimulate certain processes. First, hormone is antidiuretic hormone that stimulates the water reabsorption. And another hormone is aldosterone, which stimulates sodium reabsorption. It's important to understand that these two main components of the body fluids, sodium and water, are regulated separately, independently of each other. Therefore, it enables us to maintain constancy of the ratio of sodium and water, and a main index that shows that depends on this ratio is the blood osmotic pressure. Blood osmotic pressure remains constant while the fluid that is excreted through nephrons of kidney can be of highly variable osmotic pressure. It can be highly hypotonic and can be highly hypertonic, depending on necessity. And only due to this, the constancy of osmotic pressure of blood is maintained. First to be considered is antidiuretic hormone, or vasopressin. It's convenient to remember both names of this hormone because it has two main effects, different effects, that are achieved through different types of receptors. Hormone is produced in hypothalamus, in supraoptic mainly and periventricular nuclei, but it is secreted from the other part of brain because the neurons of these nuclei send their axons down to the posterior pituitary gland. And finally, the hormone is secreted into blood exactly within the posterior pituitary. Here we see supraoptic neurons and periventricular. The main stimuli that stimulate secretion of vasopressin are increase of blood osmolarity, which is detected by osmoreceptors of hypothalamus. They respond to the osmotic pressure proportionally to its level and correspondingly proportionally stimulate secretion of antidiuretic hormone. This factor, stimulating factor, is number one for secretion of antidiuretic hormone. And cells secreting ADH are most sensitive to exactly higher level of osmotic pressure, increase of the osmotic blood pressure. The second factor is decrease of blood volume, and bioreceptors influence these cells and secretion changes of antidiuretic hormone by decreased blood volume. Action of ADH in its physiological levels produces water retention in the body, which is achieved through stimulation of receptors in the distal parts of kidney nephrons, where water reabsorption occurs. Thus, both blood pressure and blood osmotic pressure and blood volume are restored. Water reabsorption produces dilution of body fluids so that osmotic pressure, increased earlier, becomes now closer to normal, fully restored back to normal. And water reabsorption also contributes to restoration of blood volume and finally blood pressure. But in higher doses, ADH produces the other effect due to stimulation of its V1, vasopressin 1 receptors, which are located in the smooth muscles of vascular wall. These receptors operate through phospholipase C inositol triphosphate mechanism. Inositol triphosphate formed due to action of ADH releases calcium from endoplasmic reticulum and causes smooth muscle contraction and therefore vascular constriction. But let's begin from the first event, hormone, which is the peptide, hydrophilic, dissolved in water. It cannot pass through membrane inside the cell, therefore it needs the external membrane receptor. This is 7 transmembrane receptors, which is coupled to G protein. ADH binds to the receptor and it results in activation of G protein inside, GQ protein in particular. GQ protein activates phospholipase C when its alpha subunit dissociates and activates phospholipase C. Phospholipase C activated produces breakdown of the membrane phospholipid, phosphatidylinositol biphosphate, and it becomes split into 2 second messengers. One is diacylglycerol, which activates protein kinase C, which also contributes to the final effect, but generally it's enough to mention that inositol triphosphate is another second messenger produced by activation of this pathway. And inositol triphosphate is the ligand for ligand-gated channels for calcium in the endoplasmic reticulum. So when it binds, it opens the channels and calcium is released into the cytoplasm, where, as you remember, calcium binds to calmodulin, and calcium-calmodulin complex activates myosin light chain kinase, which in turn phosphorylates the myosin heads, and it finally initiates the process of contraction, or in most cases not simply initiates but increases the degree of smooth muscles contraction in a vascular wall. So vessels become constricted stronger than usual. And systemic vasoconstriction in arterioles of the body results in increase of blood pressure back to normal. And this happens usually in response to decrease of blood pressure and blood volume. Other type of receptors is V2 receptors, vasopressin 2, which are located in kidney. These receptors operate through adenylate cyclase cyclic AMP mechanism. This is a principal cell portion. Principal cells are present in the late distal tubules and collecting ducts. Plasma membranes of these cells are entirely impermeable for water in the absence of vasopressin. No ADH, no water permeability. These cells have specific aquaporins, in particular aquaporins 2, which are internalized, and they can be inserted into plasma membrane, but insertion of aquaporins is exactly controlled by ADH, antidiuretic hormone. Let's start from the beginning. V2 receptors are also 7 transmembrane receptors in the cells, which are coupled to G protein. In this case, is GS, stimulating proteins. So when ADH molecule binds to the receptor, it results in activation of GS protein, which appears as dissociation of alpha subunit and activation of adenylate cyclase by this subunit. Adenylate cyclase starts to convert ATP into cyclic AMP molecule by separation of 2 phosphate groups, inorganic phosphate groups, from ATP and making the cycle shape of the molecule. And many molecules of cyclic AMP are formed. Each 4 of them are enough to bind to 1 molecule of protein kinase A. It binds to inhibitory subunits and removes the inhibition of protein kinase A, so that protein kinase A becomes active. And actually many molecules of protein kinase A become active. Protein kinases, all of them, phosphorylate some substrate proteins. Here the substrate proteins perhaps are microtubules, which contribute to the movement, translocation of aquaporins 2 and the insertion to the membrane. Aquaporins create the water channels in the membrane, and only due to these water channels, water can be reabsorbed. The reason of water reabsorption, the moving force, is osmotic pressure difference. But regardless of osmotic pressure difference existence, absence of water channels, does not permit water to be reabsorbed. So water channels amount is regulated by ADH proportional to the necessity in water reabsorption. Zero ADH, no water reabsorption at all. Moderate level of ADH, moderate amount of water reabsorption. Maximal ADH level, maximal water reabsorption, fully according to the difference in the osmotic pressures. Regulation of water excretion. Let's consider few opposite cases. First case, when osmotic pressure of blood is high and blood volume is low. In this case, the secretion of ADH is maximal, high level of ADH. So, permeability for water in the distal parts of the nephron is maximal, and water can be reabsorbed down the gradient until full balancing, and therefore the final fluid in the tubules reaches the same osmotic pressure as the osmotic pressure of the hypertonic medullary interstitium. So it's highly concentrated, more than 1000, it can be 1200, 1003, 1400 milliosmol per liter. Of course, due to great water reabsorption, the total amount of urine formed becomes decreased greatly. And now, opposite case, the osmotic pressure of blood is decreased while blood volume can be higher than normal. In this case, ADH secretion is totally suppressed and there is no ADH at all. And you can see that permeability for water is zero. So that practically from the beginning from the ascending limb of the loop of Henle until the very end of the nephron, including the collecting duct, permeability for water is absent. Therefore, water that remains in the tubular fluid is not reabsorbed and simply follows the tubules and reaches the renal pelvis and goes to ureter and therefore fills the urinary bladder. And amount of formed urine is great, but its concentration is very low because gradient is not used because of absence of water permeability. Hypotonic fluid is formed as a result. Again, let's consider these same two situations, but it's necessary to remember how it's named. When level of ADH is high, it creates this state which is called antidiuresis. Again, water is reabsorbed maximally so that it's conserved in the body. Water is reabsorbed especially in the collecting ducts where the hypertonic medullary interstitial that is filled with sodium, chlorine, potassium ions and urea as well simply attracts water down the osmotic gradient from tubules into interstitium and finally it goes into blood. So finally the osmotic pressure of the released and excreted urine is great and amount is low. Very small volume of highly concentrated urine is released and we conserve as much fluid, as much water in the body as we can so that it will contribute to dilute the osmotically active substances in body fluids and to contribute to higher volume of the body fluids. Opposite case, when ADH is not secreted at all, is called the water diuresis. Water cannot be reabsorbed beginning from the ascending part of the loop of Henle. Water remains in the lumen of tubules and it makes highly diluted content of the lumen of tubules. So concentration is very low, but volume is great. So this is water diuresis. Stimulation of ADH secretion by plasma osmolarity. There is a figure that shows that ADH level changes even when plasma osmotic pressure, blood osmotic pressure is within perfectly normal limits. As you remember, the norms of the osmotic pressure is from 280 up to 300 with the mean value 290. So it can be expressed as 290 plus minus 10 milliosmol. And you can see that lower limit 280 corresponds to very, very low, practically absent ADH. And any elevation of osmotic pressure within normal limits, within this normal range, immediately proportionally increases the secretion of ADH. And further elevation out of the limits increases even more and more. So you see, ADH directly depends on osmotic pressure. And actually, because it becomes higher and higher progressively with changes of osmotic pressure, even within normal limits, we very unusually may go out of these limits, because regulation starts within normal limits and prevents further increase of the osmotic pressure, contributes to immediate restoration of the osmotic pressure back to normal. Regulation of the extracellular fluid osmolarity, the total scheme, shows how it occurs. Osmotic pressure of the extracellular fluid. I hope you remember that blood osmotic pressure and interstitial fluid osmotic pressure is the same, because it's one and the same extracellular fluid. And blood differs from the interstitial fluid only by content of proteins. So, increase of osmotic pressure of the extracellular fluid stimulates the osmoreceptors of hypothalamus. And these osmoreceptors activate the thirst center, also in hypothalamus, and stimulate the supraoptic nuclei as well as paraventricular to increase ADH secretion. Thirst stimulates water intake, which contributes to faster restoration of osmotic pressure. Water starts to be absorbed from digestive tract, while ADH tries to conserve water that is already present in the body to limit its loss by increased water reabsorption in the distal parts of the nephron. As a result, water amount increases, and both contributes to dilution of the extracellular fluid, which decreases osmotic pressure back to normal, which is detected finally by osmoreceptors and thirst activation. Then it's removed and ADH secretion comes back to normal moderate level. In case of opposite changes, decreased extracellular fluid osmolarity. Of course, the changes are also opposite. Thirst is inhibited, ADH secretion is inhibited, and water excess is excreted, so that osmotic pressure again comes back to normal. Mechanism of maintaining the constant blood osmolarity. We can observe this on an example of drinking of 1 liter of water. It's rather big for once at the moment drinking, but for healthy person it doesn't produce serious problems. Just it increases temporarily for few next hours the urine excretion amount. Drinking of water, 1 liter. From this moment, what happens? ADH secretion is greatly inhibited. Why it's inhibited? Because first portions of water absorbed from the gastrointestinal tract first start to decrease slightly blood plasma osmotic pressure. This minimal decrease already inhibits ADH secretion, and as a result, water reabsorption is decreased. So urine flow rate or diuresis increases. Look at this blue line. It greatly increases for next few hours, while urinary solutes concentration decreases also correspondingly. And as a result, the osmotic pressure remains constant. It decreases only slightly, negligibly, and quickly is restored back. And also you may see that the amount of dissolved substances in the urine, amount, not concentration, remains practically constant. The same amount of substances that should be excreted is excreted each next hour. Just how it's achieved? By higher volume and lower concentration, and total amount remains the same. And what is most important for us, plasma osmolarity remains practically almost unchanged. If they changed negligibly. So even drinking of excessive water, within reasonable limit of course, does not disturb the constant blood osmotic pressure. And all this is achieved mainly due to ADH action. ADH deficiency. If you remember, we studied, and this produces the disease called diabetes insipidus, non-sugar diabetes. Absence of ADH results in formation of high amount of hypotonic diluted urine. And urine volume actually can exceed 15 liters daily. Loss of fluid may be up to 20 liters daily. And thirst mechanism is activated strongly. Otherwise, the person may not think about drinking the corresponding amount of fluid. And if someone does not drink the same amount that is lost, water balance is not maintained, and it will be great dehydration, which is dangerous for life. So, with the corresponding need of fluid, the same, the person should drink. If it's untreated diabetes insipidus, the person should drink the same amount of fluid. The treatment is administration of the synthetic analog of ADH, desmopressin. Aldosterone is second important hormone for regulation of kidney function. This hormone is secreted by adrenal cortex, by its glomerular most superficial zone. And in contrast to the hormones of other areas of the adrenal cortex, it's not controlled by adrenocorticotropic hormone, but it is controlled by the system, as it is a part of this system. That is abbreviation called RAAS, but total name is Renin-Angiotensin-Aldosterone System. As you see, aldosterone comes after angiotensin, so immediate stimulating factor for aldosterone secretion is angiotensin. But generally the whole system becomes activated when renin secretion increases. And for renin secretion, the main stimuli are the following: decrease of renal perfusion pressure. In other words, you may simply say that decrease of renal blood supply. Then, when it occurs, we studied that kidneys are able to maintain normal blood supply within wide limits of changes of the systemic pressure. So, real decrease of renal blood supply can occur only when decrease of the systemic blood pressure is significant. Then another stimulus is hyponatremia, decreased sodium level in blood plasma, and increased potassium level in blood plasma. These two factors can be easily remembered if you remember that final factor of the system, aldosterone, increases sodium and decreases potassium. So it's logical that initial stimulating factors to activate the system as a whole are opposite to effects of system. Secretion of renin occurs from the juxtaglomerular apparatus. It's located between two arterioles entering the glomerulus and outgoing, afferent and afferent arterial. And really the apparatus itself is formed by the distal tubule, which always passes perpendicularly to the plane of these two arterioles, and between distal tubule and these arterioles, the whole juxtaglomerular apparatus is located. And main parts of this apparatus are cells of macula densa, cells that are sensitive to level of sodium and chlorine in the distal tubule fluid, and also juxtaglomerular cells. These cells are located just around the afferent bringing arteriole, and exactly these cells are the source of renin. Renin is secreted exactly from the juxtaglomerular cells. So here it's produced, stored, and secreted in response to the stimuli. These stimulus listed on the previous slide. Now let's recall them again. It's release of renin into blood. Stimuli are decreased renal blood supply, decreased sodium level in blood, and increased potassium level in blood. Also, we can mention one more. Strong activation of the sympathetic system may stimulate secretion of renin. Total renin-angiotensin-aldosterone system activation events are as follows. Let's consider this sequence of events. We studied this, but one more time we'll repeat. Angiotensin is formed, and the angiotensin is formed from angiotensinogen, which is blood plasma protein formed by the liver. Liver produces globulins and albumins, and globulins, just with the exception of gamma globulins, produced by B lymphocytes, by plasma cells. And this globulin is alpha-2 globulin. Angiotensinogen is one of alpha-2 globulin. You remember that globulins are one of the largest blood plasma proteins. And this protein is always present in blood. But when renin is secreted from kidney, and when it appears in blood, it splits one little portion from great globulin. Just splits the peptide that contains 10 amino acids. And this peptide is angiotensin 1. It's still not active substance, but further it becomes converted to highly potent vasoconstricting agent. Conversion occurs by angiotensin converting enzyme. It splits 2 more amino acids so that only 8 of them remains, and this octapeptide is the angiotensin 2. It's very strong vasoconstricting substance. Adrenal cortex is stimulated. The glomerular superficial zone of the adrenal cortex is stimulated by angiotensin, and in response, aldosterone is secreted into blood, so that full system effects development is possible, because angiotensin vasoconstrictor, and it's rather quickly, within 20 minutes or so, produces increase of blood pressure due to total peripheral resistance. But effects of aldosterone are slow enough, and finally they produce increase in blood volume, so that it's a long-term blood pressure regulation. According to modern data, angiotensin 2 also has long-term effects, similar to aldosterone. But anyway, aldosterone effects we'll consider the main for restoration of blood volume. Let's list all the effects that occur by aldosterone in kidney. Higher sodium reabsorption and potassium secretion occurs in the distal tubules and collecting ducts, principal cells. Principal cells of these distal portions of a neuron, facing lumen side is to the left, and facing blood interstitial is right side. Aldosterone is steroid hormone, so it's dissolved easily in membrane phospholipids, and it uses intracellular receptors. It binds to cytosolic receptors, but then complex hormone receptor enters nucleus. Then it stimulates the synthesis of mRNAs, which finally activate the synthesis of new proteins. And these new proteins are the following. Number 1 is sodium-potassium ATPase of the basolateral membrane, where sodium is pumped out of the cell in direction of interstitial fluid and blood. So it simply appears immediately in the extracellular fluid. And then it becomes distributed evenly between 2 parts of the extracellular fluid, interstitial fluid and blood plasma. So sodium-potassium ATPase, this molecular device is a protein. So exactly this protein's amount increases due to aldosterone action. So the more aldosterone is secreted, the more pumps constantly function and produce greater and greater amount of sodium reabsorption. But first, sodium must enter the cell, and as sodium is charged particle, it cannot penetrate through the membrane of cell. Therefore, additional protein, which is formed also due to aldosterone action, is proteins that produce sodium channels. You remember, channels also consist of the protein walls, and these sodium channels are proteins are produced due to aldosterone action stimulating synthesis of new proteins. So these two components finally provide the reabsorption of sodium, while potassium is moved into the cell against gradient, and finally, when concentration increases, it passively leaves the cell through potassium channels into the lumen. So potassium is secreted. Its opposite, release from blood. Initially, potassium is taken from extracellular fluid and then moves into lumen. This opposite to reabsorption process, this is potassium secretion. One more time we consider Renin-Angiotensin-Aldosterone System. First stimulus is great decrease, significant decrease of the systemic blood pressure, which results in decrease of kidney blood supply and stimulates the juxtaglomerular cells in kidney. Also, decreased sodium and increased potassium stimulate kidney juxtaglomerular apparatus cells. Juxtaglomerular cells release renin. Renin converts angiotensin, actually converts not good word, because large globulin is not converted to peptide. Large globulin remains almost the same large globulin, just little piece of angiotensinogen is split from the molecules and it's angiotensin 1. Then angiotensin converting enzyme converts angiotensin 1 into angiotensin 2, which is highly potent vasoconstricting substance. Angiotensin 2 produces vasoconstriction, which is rather fast effect. And if we remember the classification of blood pressure regulation mechanism, there were fast mechanism, neural, and long-term mechanism, hormonal. But vasoconstriction produced by angiotensin is moderate, not very fast effect, not seconds, but not hours, which are necessary for aldosterone effect. So it's an intermediate mechanism, about 20 minutes. For development of vasoconstriction, which immediately, rather quickly produces arterial blood pressure elevation. But angiotensin 2 stimulates secretion of aldosterone, and aldosterone increases reabsorption of sodium, and water follows sodium. Of course, water reabsorption, it can be regulated by ADH, but in many cases aldosterone and ADH operate as synergistic hormones, so that sodium reabsorption is accompanied by water reabsorption. And exactly due to this water, finally we have higher blood volume, which is rather slow effect. It cannot be accumulated so quickly. Water goes milliliter by milliliter and gradually blood volume becomes higher. Also, angiotensin 2 increases the thirst sensation, and therefore it also contributes to greater water intake. And high water intake also makes the restoration of blood volume faster. And all this contributes to restoration of arterial blood pressure, and vasoconstriction, strong vasoconstriction produced by angiotensin 2 becomes not necessary, and vessels may maintain normal vascular tone. And arterial blood pressure is restored due to blood volume, which is the long-term mechanism of restoration of blood pressure. So this is a Renin-Angiotensin-Aldosterone system. Constant blood volume is maintained due to all these mechanisms by kidney. This figure shows you that regardless even of daily intake differences, except for extremely small daily intake, which results, as you can see from the figure, may result in death. Greater and greater and greater blood volume, greater fluid intake daily still does not affect the blood volume, which remains about 5 liters approximately for average person, and only it can be lost in case of extremely low water intake. And with normal intake approximately 2.5 liters or so, depending on body weight, the blood volume is maintained at the normal range, but with certain decrease of fluid intake and increase of fluid intake, at least according to this figure, up to 8 liters daily, which is extremely high. Still, you see, blood volume remains constant, and this is performed predominantly by kidney due to regulation by the hormones. After consideration of the two main hormones, we can come to the other hormones involved into regulation, and maybe the first place among others is taken by atrial natriuretic peptide, ANP. And main effect of this hormone actually can be simply deduced from the name. Natriuresis is loss of sodium with urine. So you may conclude that this hormone contributes to less sodium reabsorption, so that sodium is lost with urine. And so it can be said that it is antagonist of aldosterone. Atrial natriuretic peptides show that it's produced by atria, and it's produced in response to higher atrial pressure and blood volume. Actually, the immediate stimulus is great stretching of atrial cells, but this great stretching occurs when blood volume is too high and atrial pressure becomes increased. So in response to strong stretching, atrial natriuretic peptide is secreted from atria predominantly. Also, it can be produced by vascular wall. Main effects, really in effects of atrial natriuretic peptides are variable, multiple, but main is decreased sodium reabsorption. In blood vessels, it produces relaxation of smooth muscle, so that the total volume is higher of the systemic vascular system. Then, kidney, it decreases sodium reabsorption and produces higher filtration rate. Actually, it's antagonistic to aldosterone. And as a result, salt and water are excreted by kidney in high amount than usual. So when atrial pressure is increased, which corresponds to high pressure level, and blood volume is high, excretion of salts and water makes the blood pressure and blood volume return back to normal. Autoregulation. There are few mechanisms of autoregulation, of constancy of renal blood flow, and therefore, which is important, glomerular filtration rate. One type of autoregulation was studied in the regulation of blood pressure. It was a myogenic mechanism, when smooth muscle cells in arterioles of glomerulus produced stronger contraction or relaxation in response to stretching. High degree of stretching produces stronger contraction, decreased stretching produces relaxation, so that it helps to maintain constant amount of blood that comes to each glomerulus. But there is one more mechanism of autoregulation which also helps to maintain constancy of renal blood flow and filtration rate. First, we just observe how the renal blood flow and glomerular filtration rate changes with arterial pressure changes. Normal arterial pressure between systolic, average between systolic and diastolic, is 100 millimeters mercury, and it corresponds to certain level of renal blood flow and glomerular filtration rate. And you can see that, maybe you remember from previously studied topics, that approximately from 80 millimeters up to 180, within this range of wide range of blood pressures, both renal blood flow and glomerular filtration rate remain practically unchanged. Of course, not entirely constant, but much less, not proportional changes, almost constant. And one more mechanism how it's achieved is discussed in the next slide, how the constancy of renal blood flow and glomerular filtration rate is maintained. This mechanism is called tubular-glomerular feedback, from tubules to glomerulus. And when it can happen, where it can happen, where tubules pass nearby the glomerulus, and exactly the distal tubule always passes near the glomerulus, where it forms the juxtaglomerular apparatus. So, but let's start first from changes of the glomerular filtration rate. Imagine that due to decreased blood supply to kidney, there is a decrease in glomerular filtration rate, which decreases the functioning of kidney, because kidney must clean out of the substances that should be excreted certain volume of blood each minute and each hour and so. And if glomerular filtration rate is decreased, it results in less functioning of the kidney. While excessive glomerular filtration rate will produce less opportunity for normal reabsorption of many necessary substances. If flow through tubules increases more than normal, the time available for reabsorption becomes less, and therefore less substances can be reabsorbed. So it will be great loss of necessary substances. But let's come back to decreased glomerular filtration rate. What happens in this case? In this case, decreased fluid flow rate is achieved in the tubules. And as a result, with slow movement of fluid, reabsorption takes place more than usual. Reabsorption becomes increased. And as a result, the concentration of main substances that come to the distal tubule is decreased, and main substances detected by macula densa of the juxtaglomerular apparatus. These main factors are sodium and chlorine. So due to high level of reabsorption, sodium and chlorine level in tubular fluid is less than normal. And cells of macula densa detect this. This is a portion, now we enlarge this portion of the... So cells of macula densa, in response to decreased sodium and chlorine, stimulate two processes. First, simply stimulate the afferent arterial dilation, which is wider bringing artery, and renin secretion. Renin is secreted into blood, and finally, not immediately, but after the number of events, sequence of events, which we discussed previously, produces constriction also for the afferent arterial. So when arterial that supplies blood to glomerulus is dilated and outflow is impeded, it results in increase of pressure and therefore higher glomerular filtration rate. So glomerular filtration rate increases back to normal, and kidney function is again normal. So tubular glomerular feedback. Now let's observe it one more time on the scheme. First, the arterial pressure should be decreased, and actually greatly decreased, so that even kidney cannot maintain automatically by autoregulation glomerular filtration rate. And glomerular hydrostatic pressure decreases, so glomerular filtration rate is also decreased. And then it results, due to greater reabsorption of sodium and chlorine, results in decreased sodium and chlorine level in the tubular fluid in the distal tubules, where cells of macula densa can feel this decreased sodium, sense this sodium and chlorine decrease. Then, in response, the cell of macula densa produce the dilation of the arterial bringing, afferent arterial, and they release renin. They stimulate the juxtaglomerular cells to release renin, which in turn produces the number of events where angiotensin at least is produced and produces higher resistance constriction of the afferent arterial outgoing from the glomerulus. And both these changes of arterials contribute to higher pressure and therefore higher glomerular filtration rate. So this is the mechanism of the tubular glomerular feedback in kidney, which is an important mechanism for maintaining the constancy of glomerular filtration rate, so that it doesn't become decreased, which decreases the kidney function performance, and it must not be increased, because in this case we cannot reabsorb all necessary substances. Let's recall more detailed juxtaglomerular apparatus structure. Cells of macula densa are sensitive to sodium and chlorine in the lumen of the distal tubule, and they influence the secretion of renin by juxtaglomerular cells. So this is a glomerulus, and it's surrounded by Bowman's capsule. It's the efferent artery which brings blood, supplies blood to glomerulus, and this is efferent that produces outflow of blood from glomerulus. And there are numerous smooth muscle cells in walls of the both arterials. Distal tubule always passes between these arterials under perpendicular to the plane so that you can see cross section. And the wall of distal tubule that faces glomerulus is composed by macula densa cells that are sensitive to sodium and chlorine. And around the efferent arterial, juxtaglomerular cells are arranged, and in response to a number of factors, among which is decreased renal blood supply, decreased sodium in blood, and increased potassium in blood, they secrete renin. And also, in case of stimulation by macula densa cells, they release renin. So that's all I wanted to say about the regulation of kidney. Thank you for your attention.